So I would like to say good morning, good afternoon, and good evening to the participants. So this is, my name is Alfredo Garzino Demo, and today actually I'm standing in I have the honor to stand in for the president of the Global Virus Network, Stan Vermund, who cannot be with us for a previous commitment. For people that have joined us that are not familiar with the Global Virus Network, the name says it is an organization that puts together scientists globally to address the problem of ongoing and emerging viral pathogens that are entering our species. So we have a mission of education, we have a mission of research, and we have a mission of advocacy. So, today we are fulfilling at least two of these missions with this webinar by Dr. Maggie Bartlett. So Dr. Maggie Bartlett and her PhD both at the University of Nebraska. And the PhD was at the University of Nebraska Medical Center. It was in immunology, pathology, and infectious diseases. Okay, and after that, she became a faculty in two places for a period of time. You know, she was both with the Navy. Medical Research Command by Biological Defense Research Directorate, and even now she's affiliated with the laboratory of one of the most proactive members of the GVN, Dr. Diane Griffin at the Johns Hopkins Bloomberg School of Public Health. And Maggie is also the program director of the Global Virus Network, which is the title that I previously held, and I think she will definitely, she's definitely bringing a lot of value in that respect. The seminar that webinar she's presenting today is extremely timely, as we are understanding something that previously was underappreciated, which is the fact that RNA viruses can persist often longer than we would like them to. And there is this is a emerging field, you know, and so I think it's very timely that we have a presentation from Dr. Bartlett. And I think hopefully there will be questions at the end, but without further ado, I will yield to Dr. Bartlett. Take it away, Maggie. Thank you.
Thanks so much for the kind introduction, Alfredo. I'm really thrilled to be part of the Global Virus Network and even more excited to talk about some of my research with you all. So let's see. There we go. Just a brief overview of what we'll be covering today. So we'll start with just a little brief background. Alfredo already touched on a lot of this, but I'm from Nebraska. For people who aren't familiar with where that is, it's the red state right in the middle of the US. And I have 5 siblings, and I really came to science out of fear. So as a child, I was diagnosed with obsessive compulsive disorder, and I was just really terrified of the things that you couldn't see but could make you sick. And so I needed to understand. And this interplay between our immune system and pathogens just really fascinated me. So I went to get a bachelor's in biotechnology, and then my doctorate, and then my postdoc with Diane Griffin. And so I'm a virologist, I'm a systems biologist, and I've worked on alpha viruses, bunyaviruses, filoviruses, of course, paramyxos, SARS-CoV-2, as I think all virologists during this time, I'm sure everyone on this call who had the opportunity to contribute to what we learned did. I work with a lot of different transcriptomic, proteomic, and single cell sequencing data. And then I think it's a fun fact that I've worked with the standard human, mice, primate samples, but also bats and sharks. Not in today's talk, but anytime I'm happy to talk about those. So I'm going to start with a tale of 3 viruses. And this really started as one virus that I was going to work on with Diane Griffin at Hopkins. So Synbis virus is an alpha virus that I had joined to look at how it's cleared from the brain. But this was during the pandemic. And so like many of us, we also added SARS-CoV-2 to our experience. And if you're going to work with Diane Griffin and you're already working on 2 viruses, you might as well work on a 3rd. And one of her other specialties, of course, is measles virus. So the platform I'm talking about today, I originally built for Synbis. We then adjusted it to work with SARS-CoV-2 and then expanded it to measles. And now we're hoping to use it on a lot of other viruses. I did not do her work justice, but this webinar that she has on the website is fantastic and is a great overview of a lot of the different work that she's done and contributed to the field. And I won't get to talk about these, but these are just a couple of our publications so far. And so while working with Diane, we really wanted to look at the immune privilege site of the brain. And so while I feel like that name doesn't give it full justice, as it's really an immune specialized zone, because there's a lot of immunology happening in the brain. It's just very different from our overall system. And it's well known that viruses can cross the blood-brain barrier. And when they get in, they can infect your neurons and kill them. And since neurons are non-renewable, that's not ideal. Other viral infections like West Nile virus, when it gets into the brain, the immune system also kills the neurons. But one of the things that Diane had figured out is that plasmablasts can cross the blood-brain barrier and secrete antibodies, the molecule that's going to neutralize the virus. And not only does it neutralize virus, but it's doing something else. It's triggering something that allows for the neurons to get rid of the virus without killing the neuron itself. And so that's really intriguing. And why do we care specifically about Synbis or alpha viruses in general? Well, the distribution is increasing. So as the climate changes, the vectors that transmit alpha viruses and other arboviruses is expanding in range. And that means more and more people are being exposed than ever before. And we don't have treatments for these. And they can cause anything from viral encephalitis, so swelling in your brain, or they can cause this arthralgic syndrome. And there's also a high number of people, depending on what virus we're talking about, that suffer from long-term symptoms. And so that's, again, another really interesting aspect of alpha viruses. And so I'm going to be talking about some data from Synbis virus. It's an alpha virus. Again, it's about 11.7 kb. It's single-stranded positive sense RNA. It has, in humans, it typically causes the arthralgia form. But in mice, it causes an age-dependent encephalomyelitis. And so that gives us a really nice model for studying what could be happening in humans during encephalitis. There are a number of antibodies that have been characterized that work in the brain, and we know that they're critical for how we get rid of the virus from the brain. And of course, as already mentioned, the viral RNA can persist after the resolution of that acute disease. And in mice, this is lifelong. And so in humans, we don't quite know yet, but it's an intriguing facet. And so that viral RNA persists. And again, this is talked about in a lot of detail in Diane's webinar, but a lot of different viruses have the capacity where their RNA hangs around after you've cleared infectious virus. And so this is just highlighting over here. It's been characterized for Synbis, Ebola, Marburg, measles, different locations. And then, of course, the interesting aspect is that while most viruses that are RNA are cleared after acute, the RNA can still be detected for a long period of time. And so that's in contrast to active infections like persistent HIV or latent ones where it'll recrudesce later in life. So thinking about chickenpox to shingles. And so these dynamics between persistence and clearance are really what we had wanted to hone in on. So you have your cells, virus infects, and then the cells have a change in their transcriptome. And so before they die, you might have a message from the transcriptome, and those are things that we wanted to find out. We also were interested in what the messaging within the cells was before viral RNA was persistent, and then if it was possible for that to be triggered again. Could recrudescence, or is it just pieces of RNA? And then ultimately, we wanted to know what happened, what was the messaging when antibody was introduced to the system. And so why would we choose single-cell sequencing to answer these questions? Well, single-cell sequencing is very much like the next level of bulk RNA sequencing, where we're looking at all of the transcripts that your genes can make. But if you think of an organ or a tissue as like a bowl of berries, when you do bulk RNA sequencing, you get a smoothie. But what if you want to know the difference between the different kind of berries? And that's what we're doing with single-cell sequencing. So we can actually break out blueberries, raspberries, strawberries. And then if you want to do even further, you might notice that there's a little bit of a difference with what's on the outside of the strawberries. That would be akin to when we're looking at the proteins on the cell's surface. And so that's what I mean by multimodal. So we're doing many different kinds of libraries together. And then again, thinking back to the cells that we want to look at, if we did bulk single-cell sequencing, we'll get some of the message. But if one population of cells has something upregulated and the other has one that's downregulated, we wouldn't see that picture. So the approach that we did is through 10x. And so these have little antibodies that you can use for cell surface protein, and then they have a little oligo tag. And so the antibody can then bind the protein that's on the cell surface, and then you'll generate your gems, which are just emulsions of oil. And so you'll have one pool that has a cell and then everything that you need for labeling. cDNA synthesis occurs within each gem, and then you can generate a library and sequence it and cluster. And this is all summarizing what ended up being a truly Herculean effort by everybody in the lab. But we wanted to look at the 5' gene expression, so total transcriptome. We wanted to look at VDJs for BCRs, TCRs, the cell surface proteins, and then we also wanted to do a viral enrichment. And part of this came up because we were in the pandemic, and we wanted to make sure we were getting every single thing we could out of every study we were doing. And so this is kind of an a la carte process. So you wouldn't have to do all 5 of these libraries if you didn't want to or you didn't have the funding, but it doesn't hurt to do all of them because it's just more information that you have. And so we had titled this method EffectiveSeq. So everything from every cell, tissue, and virus, single-cell sequencing. And this was really led by me and Dr. Jessica Rubens, or pictured here while we were working on it. And so again, it's these 5 different libraries that we create, and then we integrate them all. And unfortunately, I won't get to talk about each of the pieces because of time today. And so I'm just going to focus on some of the viral enrichment pieces. So being able to call viral or virus positive cells was trickier than first anticipated. And part of that has to come from the number of cells and the transcripts of the host versus the transcripts of the virus. So we did a enrichment library for Synvis virus, measles virus. Ultimately, we made a panel for all viruses we'd be interested in. And then we can validate those reads using BLAST. We can extract the cells of interest and then confirm that they're going to multiple parts of a viral genome and then reintegrate that data. And then you can highlight within these different single cells which ones had viral RNA in them. And then you can say between these, we know that we have virus positive cells. This is some data from mouse PBMCs during infection. So you can see there are T cells that are infected, monocytes, B cells, which is pretty typical. And then you're able to break those out, what percentage of those cells, what percentage of the positive cells end up being these different subsets out of the total cells. And then you can get even further. So you can break down between cells that have virus in them to cells that don't have virus in them within these different subdivisions. So for instance, if we're looking at CD8 cells to CD4 cells, B cells and monocytes that had virus or didn't, you can see that regulation of metabolism was an important pathway for CD4, but you'll notice that for B cells, it was downregulated. So green is down, purple is up, and CD4 was upregulated. And so if we did bulk sequencing for this, that message would be missed. And same thing for some of these others, necroptosis, which is a different type of death pathway, is really upregulated in B cells. And so that's really intriguing, but not so much in the other subpopulations. And then again, we're seeing this strong contrast between B cells and CD4 cells. And so this, again, could be something that we missed before, but now we're able to appreciate in these different subpopulations. And then finally, again, with apoptosis, so infection is triggering different cell death pathways, essentially in different cell populations. And so I'm just going to go a little bit into how we might leverage this for biomarker discovery. So really, a biomarker is just a molecule, a protein, something that we can use to identify a disease. So in the case of viral encephalitis, we don't really have a good diagnostic because a lot of the symptoms are shared. So the differential is very wide. And it's the same when you think of acute illnesses, flu-like symptoms. So the flu-like symptoms are fever and a list of different things, but they're not indicative of any specific pathogen. They're really how your immune response is mounting to the virus itself. And so what we want to do is find out what kind of a marker could we detect in the blood to help identify viral encephalitis, because treatment is critical for a good outcome. And currently we don't have ideal treatments either for these. And so right now you would either have to have a CSF, a lumbar puncture to look for proteins, which is not a very fun procedure. And again, we're trying to find something that can be diagnosed rapidly. Imaging is the other gold standard for this. And so we're really balancing this method of neuroprotection, so the brain on the left, and neurotoxicity, so when you have a viral infection. And so during that process, you can see the different cells in the brain on top, and then there are the cells that are the blood-brain barrier, and then the blood beneath. And during neurotoxicity for viral encephalitis, things that are in your brain can leak into your blood. And so we're looking for specifically proteins that are from the brain that end up in the blood. And that could be indicative of disease. And then we also want to take it a step further and ultimately for individuals who end up developing a post-viral syndrome, can we predict that? So we have data from people who end up with a acute viral illness, and then a year down the line are diagnosed with a post-viral disease. And so what we're really looking at in this data is the ones that pop up at the same time of viremia. So there are different biomarkers, and that can be any of the things that we're looking at. But as your virus titer peaks up, the biomarkers that peak up with it and then decrease are the ones that are likely going to leach out. And the ones that end up being kind of a flat line are unlikely to be the ones that leach out. And then we can test for those. And so ultimately we end up with a big list of different potentials. And so the ideal ones, at least in our opinion, is ones that are already FDA approved for some other indication. So that's going to make it more readily accessible. And then again, they should be rapid and have a progression, because the intervention time is the key to having an improved outcome. And then also the diagnostics will allow for therapeutics to be tested. So we need a good diagnostic for acute viral encephalitis from alpha viruses, but we also need one that correlates to long-term disease. And so I'm just going to walk through some of the different things that we learned from this method. So we have our proof of concept is complete. We ended up using it on non-human primate, human, mouse cells, and then we did the 5 different libraries. We tested this not only on brain and PBMCs, but spleen and lymph nodes. So I didn't get to share the spleen or lymph node. And that's in PrEP. And the RNA positive cells are expressing pathways that differ based on their cell type. So that was very important for us to identify. And then the pathways that are shared are not only distinct to different viral infections, but there are some that are shared across viral families. So we did some comparison of measles infected cells versus Symbis infected cells. We have some additional viral infected cells that we're looking at now to integrate. And then really this method was to interrogate the roles of the host and the virus in the case of persistent viral RNA. And so we have mice out to day 30, and we're able to characterize the percentage of the genome within individual cells and which cells does the viral RNA persist. So while it targets primarily motor neurons, what we're seeing makes sense in that at day 30, it's mostly mature neurons. And so we're piecing out the specific subsets there. Ultimately, we've expanded our panel so that our panel can detect pretty much any human pathogenic virus within single cells. And we'd like to increase the efficacy of it so we can look at the dynamics of the viral kinetics. So we want it to be able to look at the specific mutations in different cell types if they're selected over time. And then, of course, I didn't get to tell you any of the B and T cell repertoire data, but there's some really interesting pieces of being able to identify the B cell clones and the T cell clones that actually end up in the brain during infection. And so then from that, we can clone those out and determine what their efficacy is. And then we didn't get to do this piece, but the next thing that we would be able to add in is the epigenetic library. And so that would also add to what we're getting from every sample. And so I'm program director and I'm also faculty at Hopkins. And so my goals are hopefully to get single cell sequence anywhere. And so there are some really cool technologies that would allow us to, whether it's in containment or in austere environments, be able to collect samples and make it easier for us to do this from any location. And then get to that single virus genome in individual cells, because I think there's some interesting dynamics there about how the virus is able to persist, or the RNA itself, because we don't have a good understanding of why it stays around. And then there are some interesting links between collagen and connective tissue disease on viral disease severity. And so I'd like to parse that out and then use this omics-fueled biomarker pipeline so we can kind of center all of these on the mechanisms of viral RNA persistence and clearance. And so these would come from different mammalian samples, insect samples, and then also taking a closer look at archive data now that we have a better understanding of some of these different things. And so I'm just gonna set a primer for some of the post-viral syndrome data that should be up and coming. We have a couple of really cool projects. David Sullivan had a cohort that he treated with convalescent plasma during the pandemic. And we have a really robust set of people who were followed for 1 day, 14 days, 28, 90. And then we have data from a year later, whether or not they ended up with PASC or post-acute SARS-CoV-2, also called long COVID. And so we're using the same approach on that cohort and trying to parse differences between people who go on to develop PASC and people who don't. And then we also have a project that's working on single cell sequencing for people with connective tissue diseases who end up developing PASC. And part of that's because clinically they noticed that a lot of the people who were being diagnosed fell into this connective tissue disease category. And what they also noticed was they weren't at an increased risk of death, they were at a lower risk. And so there's something particularly interesting there for the pathophysiology of that post-viral syndrome. And with that, I would love to thank everybody at GVN who's been very welcoming, Alfredo, who passed the torch fantastically, and I'm very grateful for his guidance. And then, of course, Dr. Diane Griffin, who was a phenomenal mentor and continues to be, and is an incredible human in general. And then everybody in the laboratory who helped, and the funding that provided, the capacity to do these things. And then, of course, all of the samples, the humans and bats, and all of the students and people who helped when we were coordinating these, because they were really pretty extensive freezer parties at some times. So with that, I'll go ahead and just plug that if you were sad you didn't get to hear about bats today, I am going to talk about bats and filoviruses in the hemorrhagic fever watch group tomorrow. So if you email, you can join that talk. And I'll take any questions that people have.
So thank you very much, Maggie. That was outstanding. And I'd like to start with the comment, which is the incredible value that you bring to the GVN Central, because this goes to show that the GVN, administration is not just an administration. There are capable scientists that can interface with all of our centers of excellence, and that together with the team, I think that really brings a fantastic value, so that people that will reach out to the GVN, will always know that there is a deep understanding of the question, the needs, and all of that. So thank you for that. That was the comment, and I thought that was really great that you ended with the note on post-acute sequelae of COVID, which is a very current topic. So I guess recently, actually yesterday, I went to a journal club where we really touched on these aspects of PASC, and some of the questions that are really, coming to the surface are, and I don't know if you can answer that question with what you have right now, or if you don't want to, preempt some data that you may have. But can you comment on in general on what do we know about the status of the RNA? Because when I do know that there are a lot of papers that show that there is persistent nucleic acid signal in people that previously had acute COVID. But the question is always, but what is the RNA? What is the status? I understand that from many GVN talks that it's very difficult to isolate replicating virus from patients with PASC. But what can you comment on that? What is the status of that RNA, please?
No, again, that's a great question, and it's one that's still being pursued actively. And actually, Diane mentioned there's like a article from I'm blanking on the year, but it's more than, I think it was in the 1950s, where they postulated that viral RNA stays around, and no one fully understands how or why, because each virus that has been looked at that has it has a different story. So Ebola and Marburg, right, can recrudesce and come back, and you can reinfect a year later. And we haven't seen that for some of these others that we know the viral RNA is still detectable by PCR. And part of that, I think, is the power of this approach is we'd like to build it out so that we can tell the difference between replicating virus and not replicating, and also add the spatial element, because we don't really know where the RNA is hiding. So we can do PCR on a bulk of tissue, but we don't know where it would exist and why it's not being taken out of the cytoplasm, because it should be. And so it's a good question that we're still working on. And I do see there are a couple questions in the chat. Yes. So thanks, Jonathan. So he's asking, it wasn't clear that the technique was sufficiently sensitive and quantitative so you can distinguish which cell type in which viral replication is efficient from those in which it's poor. So fantastic question. this is part of why we added on the enrichment portion. So when we first did this, we were able to get a handful of cells, but we weren't able to distinguish which ones had a lot of viral RNA versus ones that didn't have very much. So we wanted to get that integrated in. And so now we have a spectrum. So you might expect some cells have a lot of viral RNA that we can detect and some don't have very much. And then, like I said, if we add in the spatial element, you'll be able to see the pattern of infectivity. And so we're working on some algorithms now so that we can break that apart better. And then Rachel asked, what do we know about how and where the viral RNA persists even after the virus is gone? Yeah, so for different viruses, there's different stories. So we know that some of measles virus can persist in like the lymph nodes. And we know for like the filoviruses, they can persist in like the testes. But for the ones like SARS-CoV-2, where we know we can detect it nucleic acid wise for a long period of time, or alpha viruses, we don't know where it's persisting, even after it's no longer replicating. There are some hypotheses. So again, some people think that it's kept in a different location. So perhaps it isn't necessarily in the cytoplasm, but there hasn't been convincing evidence that it's able to get into the nucleus and how it would stay there. And others have put through the hypothesis, and one that I'm keen on is it's packaged in a vesicle of some kind and is similar to parasites that have two forms. So they have an acute form, and then when the immune system gets wise, they can form a cyst. And it's a very different system, but there's some very cool data coming out of UNMC where they track different viruses within exosomes and vesicles. And so that might be an interesting place that they could be existing. And then Anne says, I can imagine 3 different types of RNA, fragments of RNA that require replication to persist. That might be defective virus or subgenomic replicon-like virus, or even infectious virus. And are you able to distinguish between these possibilities for SARS-CoV-2? Again, another phenomenal question. So I didn't get to talk about it today, but I have some very cool defective viral genome data that we're hopefully submitting very soon. But I think that's an excellent point. We don't have a good understanding of what those pieces are in general, and perhaps they persist because the immune system doesn't need to get rid of them. But we're not really able to distinguish between those for SARS-CoV-2 at this point because, well, we didn't have enough samples at the time that were PCR positive. But now, so we just got the data back for 96 samples. So that's from the big convalescent plasma study. And so I'm hopeful that maybe the next time I do a follow-up, I'll be able to tell you more about the SARS-CoV-2 kinetics. And then on the other hand, there could be small RNA that doesn't require a replicase. Yeah, again, there's certainly pieces that are detected, but maybe don't require a replicase. We are able to detect really small fragments with this. And so anything that's greater than 50 base pairs, we would be able to pull through. And so I would think yes. And that's one of the things that I'd like to, when I say we want to get to be able to do a full genome, when we have that, we can also integrate in the different pieces in which there might be gaps, or perhaps we're going to see after enrichment that all we see is one region that's being kept around. And that would be interesting as well. I'm loving these questions.
Okay. I had, as you were talking, I thought about a couple of other things. That one is, you know, some of the damage that happens in post-acute sequelae. I'm not talking about any particular virus, but are due to, oftentimes are not due necessarily to infected cells, but to inflammatory cells that are triggered by, you know, so that your methodology would probably be able to discern some of the damage that is occurring in that space, right? Say when there is an intermediate cell, like an activated macrophage, for example, that's damaged.
So that's one of the other interesting things, and why I think spatial single cell sequencing will be an interesting next step for, obviously not for the blood samples, but for some of the brain samples. But you're able to see the different spectrum of cells that are impacted. So the bystander cells, if you will, that aren't infected, but you see big changes to their profile. And so those could add in, again, another interesting avenue for us to leverage for either a biomarker, or perhaps they're part of the pathology of disease. And then we can target those.
Yeah.
Let's see. Oh,
Jonathan Eubank has a new question, a follow-up question.
Excellent. Some of the spatial techniques are based on the assumption that neighboring cells have the most similar transcription profile. Is this going to be compromised if infection greatly alters host transcription, so heavily infected cells would not appear in the right place? Yeah, so, and this is a good one as well, because we have thought about that a lot. And one of the things that we learned with the different types of samples we had is that the first step is typically the software cell ranger, but it gets rid of cells that have virus in them because they don't look right, because they're infected. And so we had to figure out how to recover those and what parameters to change to make sure we were getting the right ones. But again, similarly, there are some algorithms that can allow us to fix them. For the most part, they're not altered enough that it puts them in totally different categories. But it's something that we have to be really cognizant of.
Okay. Oh, I don't see any more question in the Q&A. I was wondering also, since we're talking about central nervous system, the relevance, because many of the mediators were potential biomarkers that we could find for something that is CNS could be neurotransmitters.
Yeah.
So I guess for that, we will have to look at the genes that precise to the metabolism of these neurotransmitters to address them, right? Okay.
Yeah, and unfortunately, there was some data that I would have loved to share, but it hasn't gone through clearance yet. But hopefully that'll be out soon. But we have identified some of those biomarkers that I alluded to, specifically for alpha virus encephalitis. And so again, I'm really keen for that to pass all of the checks before it's out.
Okay, thank you. One more question from Anne Huang.
Some viruses have high cytopathicity in some cells and lyse the cells. Will you be able to capture cells just before lysis? Again, just a great question, because that's something that we were trying to track early on, because we had these longitudinal samples where we wanted to make sure we were getting the right snapshot of the cells, because ultimately even each of these experiments is going to have a different amount of cells. You typically say how many you want to target, but we tell them, for what we're looking at, we want to have 5,000 to 10,000 cells captured. And so depending on when those are collected, we might capture some of them, but we really have to modulate that to each different viral case. So if we're looking, yes, exactly, looking for co-expression of apoptosis, absolutely. So apoptosis is easily one of the most upregulated pathways in any of the data sets we've looked at for cells that have virus in them. And so what's interesting is looking at the ones that have that as a gradient. So cells that have virus but don't have apoptosis markers yet, and then the amount of viral RNA that we detect versus ones that have highly regulated apoptosis markers and the amount of RNA that they have. Oh, yeah. I think so too.
Probably some of these data would also have to be looked in the light if there is a suitable animal model, because probably an animal model that recapitulates well the human pathology, which we don't have for many viruses, but when you have it, you can probably do a lot of interesting experiments, and then look at the human to see whether, because in the human probably all you can do is some before and after. You cannot follow people.
Yes.
And so probably the acuity of the system would also depend on if you have good animal model that allows you to do some groundwork first. That.
Yeah, and that's.
I agree with the comment that this is really cool.
Right. Yeah, I was certainly had the benefit of being in Diane's lab, and she had a great model for alpha virus in mice and a great model for measles in monkeys. And then we were also at a place where we were constantly getting samples for the SARS-CoV-2 studies. And so I was very fortunate to be where I was, and fortunate to have a PI who would let me say, I want to get everything from every cell. And we just really we took it as far as we could go. Well, not really. I think we can add in the epigenetic piece, but it's a tremendous amount of data to pile through.
Again, I'll reiterate what I said before. This brings a lot of value, to the ability of the GVN to interface with scientists. You mentioned that you will be giving another presentation tomorrow with the hemorrhagic fever viruses group. Is that what?
Yes.
So I will just people that would like to participate to the meeting should refer to Ruby Ferrari on the GVN team to be able to participate to that meeting. I will be there, and since there are no more questions, I would just like to once more thank you, Maggie, for that great presentation, and everybody for their attendance and question that really helped understanding the topic even better.
Thanks so much, Alfredo, and thank you everybody for participating and being here today.
And goodbye from the Global Virus Network.
We'll see you next time.